Hello everyone. In this lecture, we are going to study about, this is the part 2 of adaptive immunity. And in this lecture, we are going to study about the major histocompatibility complex molecules. And also, we'll discuss a little bit about the cytokines. Let us first discuss about the MHC molecules. We have already discussed a lot about the MHC molecules in our previous lectures. We have discussed that there are two important MHC molecules. What is the primary function? The primary function of MHC molecules is to present peptide antigens to the T lymphocytes. We have discussed that we have primarily two types of MHC molecules. One is the MHC class 1 molecule and another one is MHC class 2 molecule. The MHC class 1 molecule, it is present on all the nucleated cells. It is present on all nucleated cells and platelets. However, these are not present on RBCs. We have discussed that RBCs and platelets, they do not have nucleus. However, platelets will still have MHC class 1 molecule, but RBCs will not have MHC class 1 molecule. This MHC class 1 molecule, this is going to present intracellularly produced peptide antigens to CD8 positive T lymphocytes. Okay, so MHC class 1 molecule will present intracellularly produced peptide antigens to CD8 positive T lymphocytes. However, MHC class 2 molecules, which are present on antigen presenting cells, these are present only on antigen presenting cells. These MHC class 2 molecules will present peptide antigens which are coming from extracellular space, and they will present the extracellular antigens to the CD4 positive T lymphocytes. Okay, so that is why CD8 positive T lymphocytes are MHC class 1 restricted and CD4 positive T lymphocytes are MHC class 2 restricted. Okay, and if you see the structure of MHC class 1 molecule, it has two chains of unequal length. The longer chain is alpha chain, which has 3 domains: alpha 1, alpha 2, and alpha 3 domains. And the shorter chain is beta 2 microglobulin. Beta 2 microglobulin. And you can see the antigen binding groove is present between the alpha 1 and alpha 2 domains. Okay, so antigen binding groove in MHC class 1 molecule is present between alpha 1 and alpha 2 domains. One more concept we need to understand now is this MHC class 1 molecule should be able to present different types of peptide antigens to T lymphocytes. That is why this antigen binding groove should be polymorphic. That means in a particular cell, different MHC molecules should have different types of antigen binding groove so that they will be able to present different types of antigens to the T lymphocytes. Okay, so that is why the alpha 1 and alpha 2 domains of MHC class 1 molecule, these are polymorphic in nature. Okay, so alpha 1 and alpha 2, they are polymorphic in nature. However, the alpha 3 domain, which is not participating in the antigen, in forming the antigen binding group. Similarly, beta 2 microglobulin also is not participating in the formation of antigen binding group. These two molecules, these two domains, alpha 3 domain and the beta 2 microglobulin domain, these two are non-polymorphic. That means they are same in an individual. The structure of alpha 3 molecule and the beta 2 microglobulin molecule in all the MHC class 1 molecules, they are same. However, alpha 1 and alpha 2, they are different. That is why they are polymorphic. Okay. So alpha 3 and beta 2 microglobulin, these are non-polymorphic. So what is the importance of alpha 3 molecule? Alpha 3 domain. Alpha 3 domain is important because the CD8, co-receptor of CD8 positive T lymphocyte is going to bind to the alpha 3 molecule. Okay, we'll discuss it again in the next slide. And if you see the MHC class 2 molecule, the structure, it is also made up of two chains, but these two chains are of equal length. Alpha chain and beta chain. Alpha chain again has two domains, alpha 1 and alpha 2. Beta chain has two domains, beta 1 and beta 2. Here the antigen binding groove is present between alpha 1 and beta 1. Okay, and for MHC class 2 molecule, the CD4 co-receptor of CD4 positive T lymphocyte is going to bind the beta 2 domain of the MHC class 2 molecule. I'm discussing all these things slightly faster because all these things we have discussed previously multiple times. Okay, so this is the basic difference between the the MHC class 1 molecule and the MHC class 2 molecule. MHC class 1 molecule, they are present on all the nucleated cells and the platelets. MHC class 2 molecule, they are present on antigen presenting cells, and the important antigen presenting cells include the dendritic cells, the macrophages, and the B lymphocytes. Of course, the antigen presenting cells will also have MHC class 1 molecule because they are all nucleated. Along with the MHC class 1, they will also have MHC class 2 molecule. Okay. Next thing is we'll try to understand the genes which are responsible for this production of MHC molecules. So basically, okay, but before that, first of all, we'll try to understand the name itself. So what do you mean by major histocompatibility complex? So it is found that if two individuals have identical sets of MHC molecules, and if you're doing transplantation between those two individuals, especially the bone marrow transplantation, there is not going to be any kind of tissue rejection. Okay. So if two individuals have identical sets of MHC molecules, if you do transplantation between them, there will be no tissue rejection. However, if two individuals do not have identical MHC molecules, and if you do transplantation between them, there will be tissue rejection. So that is why MHC molecules are responsible for the tissue rejection between two individuals. That is why the name major histocompatibility complex. In humans, initially, they found these MHC molecules on the leukocytes. That is why they named it as human leukocyte antigens or HLA molecules. But later on, now we have understood that these molecules, these MHC molecules, they are also present on various other cell types. Okay, but more or less, MHC molecules and HLA molecules, they mean the same thing. However, some authors do differ in this understanding. Some authors believe that HLA is the name that is given to the genes of the, HLA is the name that is given to the gene, and MHC is the product of that gene. Okay, anyway, we are not going to go into those controversies. For the timing, we'll just remember that more or less MHC and HLA, they mean the same thing. And these MHC molecules, these are products of genes which are present in chromosome 6p. Of course, we all have a pair of chromosome 6. One is coming from the father, other is coming from the mother. And in the short arm of chromosome 6, we'll have the genes responsible for the MHC molecules. Okay. And if you enlarge that area, we are going to find different types of genes. These are named as HLA-A, HLA-B, and HLA-C. These HLA-A, B, and C genes, they are going to give rise to the MHC class 1 molecule. But we need to understand that these genes, these HLA-A, B, and C, they will give rise to the alpha chain of MHC class 1 molecule, not the beta-2 microglobulin. Okay. So from where the beta-2 microglobulin is coming? So beta-2 microglobulin, this will come from, this is going to come from a different gene. This will come from a different gene which is present on chromosome 15. Okay. So beta-2 microglobulin is a product of different gene which is present on chromosome 15. Alpha chain is the product of MHC class 1 genes. Those are HLA-A, B, and C. Okay. Then coming to the MHC class 2 reason, basically the MHC class 2 molecules, these are product of the genes that are named as HLA-DP, HLA-DQ, and HLA-DR. Okay. So product of these genes, they are going to produce the MHC class 2 molecules. Okay. Of course, we all have different types of these genes. That means we all have different types of HLA-A molecules, different types of HLA-B molecules, HLA-C molecules, HLA-DP molecule, DQ molecule, DR molecule. That is why we all have different types of MHC molecules. Okay, we all have different types of MHC molecules. Okay, and one set of these MHC molecules come from the father, other set is coming from the mother. Okay, we'll again come to that in subsequent slides, but for the timing, you remember the MHC class 1 region include HLA-A, B, and C region. MHC class 2 genes include HLA-DP, DQ, and DR. There are some other genes which will give rise to MHC class 3 molecules. These are include the complement proteins, some of the complement proteins. However, this is not I mean, we are not going to discuss on this because this is not important for this lecture. And there are some genes present in between these MHC genes. Those I mean, product of those genes include tumor necrosis factor and leukotrienes. Again, this is not important for this lecture. Okay, so what we need to remember is the MHC class 1 genes include HLA A, B, and C, and MHC class 2 genes include HLA DP, DQ, DR, and we all have different combinations of A, B, and C molecule, DP, DQ, DR molecules. That is why we all have different sets of MHC molecules. Okay, so now that we have understood the structure of MHC class 1 and MHC class 2, now we'll discuss about their interaction with the T lymphocytes. Okay, again, these things we have discussed previously. I'll just recap everything in a slightly faster manner. So class 1 MHC molecule, it is going to present peptide antigen which is produced intracellularly to CD8 positive T lymphocyte. Okay, so if you see here, this is a protein which is produced intracellularly. This protein has to be partially digested with the help of proteasome. After partial digestion, it is going to produce small peptide fragments. These peptide fragments will be taken up into the endoplasmic reticulum with the help of a protein that is known as TAP protein. TAP means transporter associated with antigen processing. Okay, so transporter associated with antigen processing. This protein will take up this peptide antigens into the endoplasmic reticulum, and inside the endoplasmic reticulum, these peptide fragments, they are going to bind to the MHC class 1 molecules, and that complex will come to the surface. That complex will come to the surface, and now they are ready to present the antigen to the CD8 positive T cell. Okay, so this peptide antigen will be recognized by the CD8 positive T cell with the help of the T cell receptor. Of course, it is the alpha beta T cell receptor, as we have discussed in the previous lecture. Once they identify this antigen, now the co-receptor will come into play. The important co-receptor is CD8, the CD8 molecule. Now this CD8 molecule, CD8 co-receptor, it is going to bind to the alpha 3 domain of the MHC class 1 molecule. And this entire interaction, they will give the signal 1 to the CD8 positive T cell. There will be another interaction between the B7 molecule of the affected cell and CD28 molecule of the CD28 co-receptor of the T cell. B7, otherwise known as the CD80/86. Okay, so this interaction will give rise to the signal 2 to the CD8 positive T cell. Now the CD8 positive T cell is activated. Now you can tell that these are cytotoxic T lymphocytes. Okay, now these cells, these activated cytotoxic T lymphocytes, they are going to produce various products. Especially, they are going to produce the porphyrins and granzymes. Porphyrins and granzymes, and they will cause apoptosis of affected cell. Basically, the porphyrins, they will make pores in this affected cell, and the granzymes, these granzymes will go into the affected cell and they will cause apoptosis. These cytotoxic T lymphocytes, they can also cause apoptosis of the affected cell with the help of this fast ligand fast receptor interaction. Again, this we have discussed when I discussed about the extrinsic pathway or the death receptor pathway of the apoptosis. Okay, so this cytotoxic T lymphocyte, they can produce this fast ligand, which can be recognized by the fast receptor of the affected cell. Affected cell means we are discussing about this cell, of course, the cell which is expressing the peptide antigen. And through this process, they can cause apoptosis. Also, sometimes these activated T lymphocytes, they are going to produce some of the cytokines. One of the important cytokines is interferon gamma, which is going to activate the macrophages. And these macrophages can also help the T lymphocytes in clearing the affected cells. Okay. So basically, this happens for the cells which are affected with intracellular pathogens, especially the viruses and the cancerous cells or the tumor cells. Okay, so basically the intracellular pathogens, they are going to produce some of the proteins inside the cell which are not self proteins, and those proteins through this process can be expressed, can be presented to the cytotoxic T lymphocytes. Similarly, the tumorous cells, the cancerous cells, they will produce some of the proteins which are, of course, mutated proteins, abnormal proteins. Those proteins can be presented to the cytotoxic T lymphocytes, and through this interaction, they can identify that this cell is abnormal, this cell is either infected with some intracellular pathogen, or this cell is a tumorous cell, and they will further produce these mechanisms to cause damage to that particular cell. However, we have also discussed that because MHC class 1 molecule is present in all the nucleated cells, even the normal cells are going to produce, they are going to present self-antigens with the help of MHC class 1 molecule. However, most of the cytotoxic T lymphocytes, most of the CD8 positive T lymphocytes, they are not going to recognize the self-antigens. How is that possible? Because we have discussed, when I discussed about the T cell development and thymus, I have told you that those T lymphocytes who will develop receptors, who will develop the T cell receptor for the self-antigens, those T lymphocytes have to die inside the thymus itself. That is known as the process of negative selection, which happens inside the thymic medulla. Okay. So during the T cell development, if the T cells they develop T cell receptor for self-antigens, they will die inside the thymic medulla in the process of negative selection. But sometimes, still in the peripheral blood, you are going to find some of the CD8 positive T lymphocytes which can identify the self-antigens expressed by the MHC class 1 molecule. But even though they identify those antigens, they are not going to harm the particular cell. They are not going to harm the our own cell. That is possible because of the mechanism of immune tolerance. Okay. All those things we'll discuss when I discuss about the autoimmunity. For the timing, you remember, even though the cells expressed self-antigens with the help of MHC class 1 molecule, and even though CD8 positive T cell sometimes can identify those self-antigens, they are not going to harm the self cells. They will only harm the cells which are affected with intracellular pathogen or the tumorous cells. This is about the class 1 molecule. Now we'll discuss about the class 2 molecule. Class 2 molecules are present only on the antigen presenting cells. They will present only the extracellular antigens. Okay. So if you see here, these are some of the extracellular proteins. Of course, these are abnormal proteins, especially the bacterial products. Okay, or sometimes the viral products. These proteins, first of all, it will be taken up into the cells and they will make the endosome. Sometimes they will fuse with the lysosomes. And after this fusion, these proteins can be digested with the help of lysosomal hydrolysis, and they will make the peptide fragments. These peptide fragments can bind to the MHC class 2 molecule and they can come to the surface. Okay. Now this antigen presenting cell, with the help of MHC class 2 molecule, it can present these extracellular peptide antigens to the CD4 positive T lymphocyte. So this is the CD4 positive T lymphocyte. Okay, otherwise known as the T helper cell. Okay, and also if you see the interaction, once the T cell receptor identifies this peptide antigen, the CD4 molecule can come and bind to the beta 2 domain of the MHC class 2 molecule. And the other interaction is same, that is the B7 CD28 interaction. This will give the signal 1. The B7 CD28 interaction is going to give rise to signal 2. Both these signals will activate the CD4 positive T cell, and they are going to produce various products. One of the important cytokine is interleukin 2. So this interleukin 2 has autocrine function. So it is going to act upon the same cell, and it can cause proliferation of this T helper cell, CD4 positive T helper cell. Okay, so after this proliferation, they can differentiate into various lineages. They can differentiate into the TH1 lineage, TH2 lineage, or TH17 lineage. TH1 lineage will ultimately activate the macrophages. TH2 lineage will activate the B lymphocytes. TH17 lineage will activate the neutrophils and the monocytes. Okay, and through this process, they are going to damage the extracellular antigens or extracellular peptide antigens. Okay, so this is how the MHC class 2 molecule works. Again, these things we have discussed previously. Again, we'll discuss all those things in a separate lecture when we'll discuss in detail about the TH1, TH17 lineage. Okay, so now we have discussed about the class 1 molecule and the class 2 molecule. Now we'll discuss a little bit about the clinical significance of this MHC molecule. So if you see, again, it is the chromosome 6. One is coming from the father, another one is coming from the mother. So all of us have MHC molecules, and one set of MHC molecule is coming from the father and one set is coming from the mother. So if you do HLA matching between a person and the parent, we are always going to find haplo-identical match. That means half of the MHC molecules are matched with a particular parent. Okay. And what do you mean by haplotype? Haplotype means the set of MHC genes which are present on a particular allele. Okay. So if you take the, if you take the chromosome 6, it has, of course, it has 2 chromosomes. In one particular chromosome, whatever set of genes we have for MHC molecule, that set of gene is known as haplotype. Okay. So haplotype is nothing but set of genes on a particular allele. Okay. And I've told you, if you do HLA matching with parents, you are going to find haplo match. You will always find haplo match because only half of the, half of the MHC molecules come from one parent and the other half comes from the other parent. So with parents, we are going to have half match always. And if you do matching with the sibling, 1 out of 4 siblings will have the same HLA pattern. Okay. So 1 out of the 4 siblings will be HLA matched. Okay. So these are the numbers that you have to remember. Regarding the clinical significance, we have discussed that these molecules, they are the determinant for the tissue compatibility. We will also discuss a lot about this MHC molecules or the HLA molecules when we discuss about the autoimmune disorders. But apart from that, there are some other points that I want to discuss here. Is the first one is allergic to a particular antigen. For example, as it is given in the textbook, if a particular person has MHC molecule which can identify ragweed pollen, if a person has a particular MHC molecule which can present this particular ragweed pollen antigen, that particular individual is going to be allergic. That particular individual is going to produce, it is going to mount immune response against this pollen, and that particular individual is going to be allergic to this particular antigen. Okay. So if you take 2%, one is having MHC molecule which can present this ragweed pollen, and other person is not going, other person doesn't have the MHC molecule which can present this pollen antigen. The person who have the MHC molecule to present this antigen, that person will be allergic to this ragweed pollen. However, if the person doesn't have MHC molecule which can present this pollen antigen, that person is not going to be allergic for that particular antigen. Okay, so that is one concept. The second thing is regarding the correct immune response against different microbes. So this is actually very beautifully understood concept. As I have told you, the intracellular antigens or the intracellular microbes, this can be identified by the CD8 positive T lymphocytes. This is CD8 positive T lymphocytes with the help of MHC class 1 molecule. These are known as cytotoxic T lymphocyte. That means it is going to kill this particular cell. Of course, if the cell is affected with intracellular pathogen or if the cell is a tumor cell, the cell has to be destroyed in toto. So that is why they will activate the cytotoxic T lymphocyte, which will directly cause damage to that particular cell. However, if we have some extracellular microbes, these extracellular microbes can be damaged with the help of antibody molecules. So that is why these extracellular microbes, they are going to activate the they are going to activate the CD4 positive helper T lymphocyte, helper T lymphocytes, which is ultimately activating the macrophages, and the B lymphocytes are going to produce the plasmas. They are going to produce the antibodies. These antibodies can cause damage to this particular antigens. How is this happening? We'll discuss in detail, but now you should remember that if it is an intracellular pathogen or it's a tumorous cell, they will activate the cytotoxic T lymphocytes so that those T lymphocytes can damage the cell in toto. And if it's an extracellular microbe, they can ultimately activate the CD4 positive T helper lymphocyte, which will activate the macrophages and B lymphocytes. And those B lymphocytes, when they produce antibodies, those antibodies can actually cause damage to the extracellular pathogens or extracellular antigens. Okay, so this is how beautifully our immune system is organized. So this is about the MHC molecules. A little bit about the cytokines. Cytokines are known as messenger molecules of the immune system. So what is the definition? So these are soluble secreted proteins, soluble secreted proteins, which modulate the immune cells. Immune cells include the lymphocytes, the macrophages, the dendritic cells. So various immune cells, they can actually modulate various immune cells. So what do you mean by interleukins? Interleukins. Basically, interleukins are those cytokines which mediate the actions of leukocytes. Okay, which these are those cytokines which mediate the interaction, or we can say which can mediate the communication between the leukocytes. That is why the name interleukin. Okay. Based on the mode of the action, they can be divided into autocrine cytokines, paracrine cytokines, or autocrine, paracrine, and endocrine. Okay, so sorry. So based on the mode of action, they can be divided into autocrine cytokines. Autocrine cytokine means these cytokines, they are going to act on the same cell which is producing the cytokine, same cell. As we have discussed previously, that interleukin 2 is a autocrine cytokine which is produced from the T lymphocytes, and it is acting upon the same T lymphocyte to proliferate the cell. Paracrine cytokines, they will act upon the neighboring cell, nearby cell. And endocrine cytokine, it is going to act upon the distant cell. Okay, cytokines, they take part in innate immune response, they take part in adaptive immune response, they also take part in process of hematopoiesis, as we have discussed various colony stimulating factors like the GCSF, GMCSF. So various colony stimulating factors, those are also cytokines. They help in the process of hematopoiesis. The cytokines that are taking part in innate immunity and adaptive immune response, those things we'll discuss whenever we discuss that particular section. Okay, so these are just some of the definitions about the cytokines. We'll discuss individual cytokines or interleukins when we'll discuss that particular section. Okay, so this is about the MHC molecules and cytokines. In the next lecture, we'll discuss the overview of function of adaptive immunity.